Okay, we're back for the second lecture on antioxidants, Beyond the Hype. This is quite a lengthy lecture, so the video will be broken up into at least 3 parts, I would think. I'm going to talk about what antioxidants are, talk about some of the rubbish you can find online, talk a bit about free radicals and the issues with oxygen and why we think aging happens, talk about possible ways in which you can mitigate that, such as caloric restriction, what antioxidants are, and we'll have a look at some examples. We'll talk about the issue that antioxidants can also be pro-oxidants, and then we'll wrap up with a couple of sections on the issues around nutritional research and how we do it, which I think is quite important. So, off we go. Antioxidants. Antioxidants are of considerable interest, both scientifically and commercially. This lecture will focus on the evidence, which is complex and often contradictory. As I mentioned before, many claims are made for products which are claimed to be antioxidants, and in a rather large number of cases, these are based on spurious science at best. Antioxidants in our diet are a very good thing, hence recommendations like 5 a day. The detailed reasons why they're a good thing is often very hard to establish. Fruit and veg are a good source of antioxidants, also of other vitamins and minerals and often fibre, hence they are a good thing. This is typical of the commercial resources you might see online. As usual, in my notes, you can link through either from the notes page or images if you want to have a look at this. It has some good information, some of which we'll revisit later. For example, the browning reactions that cause apples to go brown when sliced. A cup of green tea and some chocolate, which all sounds very nice, but the claims made for the beverage are often not well supported by the evidence. We discussed resveratrol in the food colours lecture. It has beneficial effects, but they remain to be established. And chocolate, although very nice, contains lots of fats and sugars. If you want to consume resveratrol, plant-based sources are probably a better option. So if you want to read up about that again, go back to the food colour lecture. Again, this is from the resource I mentioned a bit earlier on. This is very poor advice. If you're smoking, don't take extra vitamin C, stop smoking, would be the advice from any healthcare professional. Okay, free radicals. Radicals are innocent, as we used to say in the 70s. It was funny then. Free radicals are highly reactive chemical species that are very important, if not often very well understood in detail, roles in biology. Contrary to what you may often read outside of good science, free radicals can be both a bad and a good thing, and are in fact essential to biochemistry. As we discussed before, context matters. Okay, so where do they come from? Here's some highly generic chemical reactions. A and B are not any specific element, but placeholders. We'll see some real examples soon. Free radicals are chemically very reactive species formed when a chemical bond splits such that each of the 2 new chemicals has a spare electron. Technically, this is known as a homolytic reaction, from the Greek homo, the same, and lysis, meaning splitting. Here the asterisk indicates 2 new chemical species which each have a spare electron. For comparison, here's a standard chemical reaction which are called heterolytic chemical reactions, which are much more common, hetero meaning different. Here, when the chemical bond broke, A lost an electron, and which was claimed by B, and that is what we see more often. But in some cases, we do see free radicals being generated, and they are very highly reactive indeed. Here's a real-world example, the homolytic fission of the chlorine molecule. This often happens in the upper atmosphere. Cl2 interacts with the UV photon, which causes the bond to split homolytically, producing 2 chlorine free radicals. The radicals, as I mentioned, are very reactive. Each one may react with another chlorine radical, which stops or terminates the process, but in many cases it's said to be the initiation step of an often quite lengthy process that produces many more free radicals, and in life can be quite damaging. Okay, so the process in a bit more detail. It starts off with the step we've just looked at, actually, called propagation. Here's an example from methane, so CH4 methane here, and a chlorine radical. We'll notice CH4 has a couple of bonds and a couple of electrons in that bond there. So, as I mentioned, a free radical can interact with another non-radical molecule to produce another free radical. Here a chlorine free radical reacts with methane, produce a different free radical, the methyl free radical, which is this thing here, and some HCl, hydrogen chloride gas in this case. This can continue for some time, a chain reaction that keeps producing more free radicals. You might say, but this is still just one free radical, and that is correct. However, when a free radical interacts with biological molecules such as lipids or nucleic acids, it can cause significant damage, and that's what we're concerned about. There are issues with free radicals in the upper atmosphere as well, but that's a bit outside the scope here. Okay, how free radical reactions terminate. Typically when two free radicals meet each other, so two chlorine radicals meet each other, produce a chlorine atom, a Cl2, and there's some other examples there. The reaction's easy to do. The problem is that free radicals must find each other, and they're often in an environment where there are very many fewer free radicals than non-radical molecules, so they tend to react with radical molecules, spawning more free radicals. Okay, why we care. Free radical damage to biological molecules. They cause damage to lipids, proteins and DNA. There are plenty of good resources in this if you're interested. And here we're just going to look at the main issues. If you're interested in in-depth read on these issues, see the paper from Peterson here, linked here. But bear in mind it's a very technical paper. What we notice here is damage to lipids, proteins and DNA. Also RNA probably as well. So here's an innocent lipid minding its own business. The R at the ends of the molecule are just placeholders again for the rest of the molecule, the parts of the molecule that don't take part in the reaction we're interested in. Chemists are lazy, so we don't draw the whole thing out. Here's another example where we've just drawn out the even more lazy way. We haven't even drawn the carbons and hydrogens. We've just drawn the chain, and you have to recognise that there's CH2 here and a CH there. It's a lot simpler to draw and helps avoid confusion when you're drawing complicated reactions. Carbon and hydrogens are still there, just not shown. Check that you can understand this and maybe try some examples yourself. In the second trace, I've also drawn the arrangement of the bonds more accurately. Note the effect that double bonds have on the shape of the molecule and their locations. This will be important in our discussion soon. Okay. So here a hydroxy free radical, that's the OH star thing, reacts with a lipid. It causes several changes. First, there is a radical electron on one of the carbon atoms now, over here, which means the lipid is now a free radical and reacts with other molecules, causing further damage. Crucially, note that the location of one of the double bonds has changed. It's a bit hard to show in this 2D image, but the overall shape of the lipid has changed as well, and this can have very significant health consequences if a lot of this damage takes place. We find lipids in cell membranes. The cell membrane separates the interior of a cell from its outside environment. It's composed of a phospholipid bilayer with embedded proteins. Although representations of it are usually static, it's in fact somewhat fluid, with various components moving in and around the phospholipid matrix. If you're interested, see this LibreText link for more information. But the phospholipid, as we imply, is a molecule which is a lipid, as we saw in the previous slide, which also has phosphorus groups as well, and they're subject to free radical damage. This is a phospholipid. It comprises a phosphate group to which two lipid tails are attached, these zigzaggy things. Again, haven't drawn the carbons, haven't drawn the hydrogens. This one's said to be saturated because it has no double bonds in the chain. This one is unsaturated because it has a double bond. If the unsaturated lipid undergoes free radical attack, its shape will change. If enough unsaturated lipids are damaged, this will significantly affect membrane fluidity, with potential health effects. In some cases, damage is so extensive that membranes can be ruptured. In other situations, carcinogenic, that is cancer-causing potentially, and mutagenic, potentially causing birth defects, products can be formed. That's more of an issue with DNA, which we'll see in a second, I think. Yeah, DNA. DNA is another molecule that can be damaged by free radicals. It's not particularly stable. It's quite easy to damage. There's some statistics around DNA and free radical damage. Over billions of years, mechanisms have evolved to auto-repair DNA, but they aren't perfect and damage may accumulate over time. So pause the video and have a read of that if you're interested. Yeah, so here's an example. Again, an innocent DNA-based guanine over here. It's attacked by a free radical, OH free radical, which seems to make a relatively small change to the base. It's gone from having a hydrogen over here to having an OH. However, this has changed the shape of the base, and it can no longer carry out its role in coding for proteins, with potentially very serious consequences. Of course, changing the single base is probably not significant. What matters is the effect of cumulative changes, of which more later. Yeah, okay, so there's a change there. For now, we'll pause this video and we'll come back with part 2 in a sec.